and now for something completely different. So AgeCurve, how do we deliver, trying to deliver deep age profiles via personal proteomics? First of all, I'd like to acknowledge we are very early stage. Since last year selling the kits. The whole story when we started work is roughly 1.5 years old. And I would like to acknowledge our early adopter AgeCurve users, because basically I designed this app, we designed this app to people like you who put their money to buying these kits, because we're selling it to end users where their heart and the body is, and trying to educate themselves and of these topics, of these kind of nerdy application that we built, of which they have quite a partial understanding of as of this now. But hope it's going to be a very nice learning journey for all of us. And next, so we are based in Cambridge. I'd like to acknowledge my team. But we are based in Cambridge, UK, the motherboard of SENSE, but most of our people are actually fully international on the East Coast, West Coast US, continental Europe, Sweden, York, not just Cambridge, but that's what truly Cambridge means to me, being fully international. Basically, hardly anybody knows me here, not many people. Just a little bit about myself. So when I was 14 years old and I started my first journal diary note, it was not about, hey, I have fallen in love with Anita. I might have, but it was about the mitochondrial theory of aging and how to blah, blah, blah. Something I read about it, and how to fix this, how to counteract this thing, and have a ground theory about it. Why? Because I wanted to fulfill all my capabilities, and I wanted to give this opportunity to work on this for other people as well, because we have many things in me that are not being fulfilled during our normal life expectancy. So ever since, and then this puzzle of aging. So I was born in the same city and the same year when the Rubik Cube was designed in Budapest, Hungary in the mid-70s. And so I became a mitochondrial stem cell biologist by default. That has something to do with this. And I also have a philosophy degree, and my thesis was about the moral, philosophical consequences of lifespan extension, because it's a serious issue. And then participate in the community as well. And in the last 8 years, I've been working with mass spectrometry-based proteomics and bioinformatics mainly. It's computational biology in Hinxton. It was in the European Bioinformatics Institute. And at the ripe age of 41, I finally ventured into business. By the way, once I checked that the median age of a biotech startup founder is 41. Go back to the first comprehensive view of aging. There was this guy called Aubrey in Cambridge in 2002 who first dared to put together a collective catalog of all the aging-related damages, and then coming up with engineering solutions to roughly address all of them. And those are those 7 damages around them you can see. And then mainstream molecular biogerontology came after 11 years after that, with this process-centric view, the hallmarks of aging paper. And now we have a real nice curriculum. If you put them together, it doesn't take much brain to map these different things together. Aubrey was kind enough to provide me with a comparison paper of these 2 views and the differences as well. It's very interesting. And so clocks, markers, patterns around these different aging processes are going to arise, and we already seen 2 candidates, out of which Steve was presenting the most accurate one, the epigenetic clock, which is great to have. And my estimation is that for the rest of the processes, other clock attempts are going to be coming. Or if not clocks, markers or patterns that are telling you something is accelerated or decelerated. It doesn't have to be a clock as accurate in order to be useful. And one thing to keep in mind is that these multiple clocks patterns are going to measure different processes. That's why, personally, I would like to call aging as agings, using the plural, like cancer, cancers, in myself, scientifically. Yeah, we see the big organismal thing at the end of it, and we have our intimate relationship with it in our mind, but scientifically speaking, I'm more comfortable with separate, diverse, highly interconnected processes. And Alex from In Silico mentioned this Balsky study that compared 11 clocks or age markers and concluded that these are measuring probably different things because they're not talking very nicely to each other all the time. And it was on 1,000 people. By the way, they used the middle age at 38 years of age. It was a bit youngish to me, but it's a nice cohort. So when I was looking into these process-based framework, and I have an understanding on proteins. Proteins are being the action molecules of life. So if you go to a gym, those people in there understand actin and myosin probably more than DNA. I've seen proteins here for the rest of these processes. Hey, loss of proteostasis, very integrative hallmark. Mitochondrial dysfunction, cellular senescence, immunosenescence, inflammaging, I see proteins at work because I'm biased towards proteins and we can measure them. So what we'd like to do, and what we call deep, it only means that we're already trying to cover fragments of 4 of these hallmarks there, indicated in red. And let me tell you how. But first, just tell you that our processor, AgeCurve AppFlow, how does it look like? So it's the emerging standard in DT, proteomics profiling. Personal genomics, you see, was saliva. It started like with 23andMe 12 years ago. And so users register, purchase a kit, saliva collection kit is sent to them, and then it is being hits the lab. Our lab is in the US, by the way. And it's a partner lab because we are a digital health company. And then it is being scheduled for a mass spectrometry run in the next couple of weeks. Then we get the raw data, do all of our extensive quality control procedures, and doing our computational software stuff on top. And then after the collection and the pickup, 6-7 weeks later, this is quite standard as well, we deliver these results in a native web application. So, and we are extending all the results because we have lots of data and we're constantly revising them, trying to come up with some good stuff. And then the education and the usefulness begins for the users and the courses. So currently we are selling kits only in the UK and started the US pilot recently, and it's looking good right now, and it's getting the good crowd in, the good early adopters that we need. Obviously, it's not mainstream. We are very early on. That's a big jump over there. It's risky. So we don't serve one-page PDF reports. This is a native web application. You take your data with you. You can always look at it. Currently, we are optimized to do one-time age profiles, not the actual trajectory, which is going to be the main thing. That's why we called AgeCurve, when you have a baseline, and when you are experimenting yourself with something, lifestyle, usual choices, and you want to see. The jury is out on that one, by the way, how many times you want to do this. You know, in gyms, when they do these blood tests, they do it 3 times a year, or I don't know this, actually. To do 1 time every year, every 5 years once. It's a big question to me. And now, talk about the technology. How do we achieve this? What is the fuel behind? It's mass spectrometry. It's been mentioned a couple of times in the last couple of days. Amy mentioned it as well. So you start with a complex biofluid, urine, blood, saliva, tens of thousands of different types of proteins and proteoforms in very different abundances in a very high analytical range. And you digest them towards its constituent peptides. These are amino acid sequences, 30, 40, 80 amino acid sequence long. And those are the things that we actually measure in mass spec. And then you ionize them and you send them after lots of separation processes to get more data on like retention time into the mass spec. And what you actually measure there is the mass over charge ratio and the intensity of that signal in the MS1 level. By the way, this is a really good review paper by Rudi Eberschuld and Matthias Mann from Nature 2015. And since we are here in Germany, I must say that I think mass spec was invented in Germany in many ways. So some of the semi-gods of mass spec are German native speakers. Rudi is from Switzerland, I think. Matthias Mann is definitely German. So once you have this MS1 level spectra acquired on the precursor peptide ions that you let into the machine. It's already is giving you characteristic signals to infer back the proteins that were there in the sample. But what's better than this is then you fragment these further in another mass spec run, MS2 levels, to get the little triplets of amino acids, or even some one amino acid, to get the sequence back. And that gives you way more information, much more confident. It's in the last 20 years, it became very robust. It started as something that's good for identifying, but in the last 10 years, it's been proved that quantifying it, that's almost as nice, and on a proteome-wide level. So the quantification that we use, it's a relative quantification. You're using these MS1 level intensities to compare across different groups, and that's very, very robust across of 5 order of magnitudes, so 1 in 10,000. And in terms of the saliva, there's been good studies out in the last 4 years, say, one of the Mathiasman group, on healthy subjects that have established checking all the variations of these thousands of proteins on the human proteome and the oral microbiome. I see a couple of people sleeping. Saying that a single run analysis can deliver the actual biological differences. Just one run, you don't need to do many. And another good thing about the saliva, not just this being much less invasive, it's not being invasive at all, you just spit, is that the top 15 most abundant proteins are just taking up third of all the mass proteome signals. So you can get down to rare proteins. As opposed to blood plasma, when you have these big guys like albumin, taking more than 90% of the signal. Urine, it's almost 60%, so that's a good thing. Another thing is that how you can use the saliva as a blood proxy or not, there are different results here. And in terms of actual variability, so it's an important question to ask, what is the interday or intraday variation levels? When I'm waking up and providing the sample, that's what we do to try things standard, or after toothbrushing, after eating, 2 weeks later, same condition. And what turned out to be is that most of these important proteins, and many of them that we measure, they are within a twofold difference, plus, minus, increase in a linear scale. So in order to something be useful as a biomarker, you should discriminate between two conditions or whatever. You need to be the median value, mean values being more than twofold difference. And the threshold that we apply is much higher than that. But we are not doing any clinical stuff technically. So what we do is sequence, quantify 4,000–6,000 of proteins, unique proteins, to get your molecular snapshot at one time, to deliver you your personal proteomes. And we do a 2-in-1 thing, because with the same machine, and your saliva is a host of lots of mostly harmless commensal bacteria, also lots of viruses as well. So half of our signal are coming from bacterial proteins, and we deliver those as well. That's an interesting thing I'm not going to talk about because that's not aging focused. And the human proteome, think about those 3,000 proteins, and quantify them. So that's how we are positioned. If you think other services like personal genomics or personal microbiome based on DNA identification, what we built here is not a competitive stuff. It's very complementary to those offerings. Some more technical numbers, and I don't want to bore you with them anymore. One such 1-hour global proteomic profiling assay gets you back 10,000 of analytes. If you combine them from all of the users, it's tens of thousands. If you measure modifications, it's 100,000, and that's quite good. So one of our example story that we deliver back on top to our users, besides all the nerdy, actual individual level thousands of proteins, is the proteome maintenance story. It relates to the proteostasis, the ubiquitin proteasome pathway that's very important there, to keep proteins in shape, to keep misfolded proteins away, degrade them. And one way to measure this is actual down regulation of the proteasomal proteins, the protein expression level. And that's exactly what we measure. So this is one view from our interface. So the whole idea here is that We call them protein guards. That we try to give out a score that is not nerdy with numbers, but you have like these 3 balls as a score. And 2 means what we say might indicate your typical aging because you are within close distance to the median, and we know the variation. 1 might indicate accelerated aging. 3, in case of proteasome, if you have overexpressed this very nicely, that might indicate per your age group, that might indicate decelerated aging. So we're really close to these biological processes, and it's based on a priori knowledge. It's not machine learning. When you pull together these things, obviously we do them because it's a multi-dimensional vector that we have, and those are the other stories. And this distribution, and that's what I'm called agings here, these results of how these systems are working, it's all based on biological assessment. And in some cases, like innate immunity, the other, what means decelerated is if you have less expressed, like I'm going to tell. And then you go down to the details, and in the protein guard story, we measure ubiquitins, proteasomes, and chaperones quickly. So currently we're running on seed money, and we are cash flow positive, but soon we might be ripe for early stage investment. But I'm not here to do a sales pitch like that. We are selling in the UK and US currently, but I hope that continental Europe and parts of it is coming anytime soon. That's figuring out the logistics and seeing the market demand. And please try us out, and any emails, any questions, I'm happy to take. Can I have just one more slide about something else? It's just since we are here at this Undoing Aging conference, in this community, it's just an advocacy advice for lifespan extensionists. It's what I do in Cambridge. Usually, there's a lot of small talk you need and chit-chat, and people like to talk about the weather. Instead, I usually talk about age and aging, trying to guess other people's age. It's not really offending at all. It's funny. There's the Sherlock Holmes element to it, to figure out, after talking with somebody and checking. Like Alex mentioned, we are all these deep neural networks having our patterns delivered. And so you need to go out and let people know that we have growing knowledge of aging and growing knowledge on how to potentially control it and get the control over it. And this is possible. It's within our realm, and unlike weather. And so do this with empathy, but show that if by fighting biological aging, counteracting it, we also fight at the same time chronological ageism. That is a very pervasive, big discrimination, going undetected many times. Thank you.